Hello, everyone. Good afternoon. I'm Martin Akerman. I'm CTO and co-founder at Envisagenics. I'm here with Maria Pineda. She's my co-founder. She's the CEO. We're both alumni from Cold Spring Harbor, so we're super excited being here today. Cold Spring Harbor is we are a spin-out from Cold Spring Harbor. Cold Spring is part of our company as well. We've been around for about 3 years. We're a team of 6 as of now. Our headquarters are in Midtown Manhattan. And Envisagenics has developed a technology that combines cloud computing, artificial intelligence with RNA-therapeutics and RNA-seq analysis. And all of this to solve a problem that I'm pretty sure people in this room are well aware of, and it is how expensive it is to create a new drug. It costs $2.6 billion. It takes over 10 years to bring a drug from preclinical studies to commercialization. And part of the reason is uncertainty. Sometimes we don't know if the targets are going to work. We don't know enough about them. We don't know if they will translate from the animal to the human, or we just can't drug them good enough. But most recently, with the advent of next-generation sequencing, people are using genomics data to investigate cohorts of patients, find commonalities among patients, find new disease-causing mutations, understand disease mechanism, find better drug targets, and so on. And this is all great. I mean, it's really good. It's really good news. But I can see at least one caveat with the genomic approach, and it is that even if you find a highly significant mutation, a disease-causing mutation, you still will be facing many years to figure out how to drug that mutation. So time-wise, we're more or less left in the same spot. So today I'd like to claim that RNA therapeutics can reduce the druggability gap, can make targets get drugged faster, because RNA therapeutics compounds and their characteristics can be predicted using artificial intelligence algorithms. So RNA therapeutics are chemically modified RNA-like molecules that bind to endogenous RNA and they modulate the gene function and the structure of the RNA through splicing modulation. As we can see here, the blue molecule is an endogenous, naturally occurring RNA of the cell. The RNA therapeutic is an antisense RNA that base pairs through basically Watson's Crick base pairing rules. And once we identify a good binding site for an RNA therapeutic, and here lies the challenge, right? So when we do predict a good binding site like that, we can go ahead and synthesize an antisense RNA to make it bind anywhere we choose in the transcriptome. Usually the way that these molecules operate is not the only way, but is one of the major ways, is through modulating alternative splicing. And that way we can choose different isoforms that we want to promote or demote, and we can control the expression of the genes in that way and acquire the effect that we're looking for. And this, of course, in contrast to small molecules or classic drugs, which are more complicated in the sense like three-dimensional structures, and the way they interact with proteins is more complicated. Proteins are highly complex three-dimensional structures, and sometimes to predict how a small molecule would bind a protein is hard. It's not impossible, though, but it's harder than looking at the base pairing, Watson's Crick base pairing, and going base by base. So it is a fair assumption to make in RNA therapeutics that we're looking into a two-dimensional entity, even though RNA does fold, but it's a good enough approximation to work in 2 dimensions, and that simplifies a lot. The next thing is that you can't have a small molecule to look the way you want. You have to kind of like look into the libraries that pharmaceutical companies own and see if anything in the library will bind your target, and if it doesn't, then you have to find another way or different library and so on. And this is sometimes why it takes longer to develop a small molecule. And then about the mechanism of action of these drugs, it depends. It depends on the protein. Proteins have diverse functions, so every protein might interact differently with the drug. Even though, in contrast with RNA therapeutics, we can assume a pretty standard way of looking at this interaction and this mechanism through splicing regulation. Another amazing thing about RNA therapeutics is that they work. They work pretty good. Here we see Adrian Krainer. He was my boss when I was a postdoc at Cold Spring Harbor. He's holding Emma Larson. Emma is one of the children that came to the clinical trials for nusinersen. nusinersen as RNA therapeutic drug to treat spinal muscular atrophy that was recently approved. December 2016 was approved by FDA in collaboration with Ionis and Biogen. This is very exciting. And if you guys have time, I will encourage everyone to go to YouTube and look for Emma Larson's story or any other SMA children, and you can see all the children that were helped by nusinersen and this type of approach. But yet again, we're a software company, right? So we work on the cloud, we do artificial intelligence. So how much can software do to accelerate RNA therapeutics? A lot. Why? Because of all the predictability that I've been discussing. So at Envisagenics, we work in 3 major fields to accelerate RNA therapeutics discovery. First of all, to discover new drug targets using RNA-seq data to find alternative splicing changes that can be drugged by this approach. We have built a resource called SpliceCore. It's a cloud-based platform that utilizes artificial intelligence to perform this task. The second thing is to predict the exact binding location of the RNA therapeutics by modeling the way in which RNA therapeutics will block the binding site of RNA binding proteins using ClipSeq data and similar tools to interpret the binding locations. And finally, once we have a candidate, an RNA therapeutic antisense RNA molecule, we can investigate the binding specificity of this molecule. We can see if there are any off-target effects and kind of approach the toxicity and the side effects of this protein, of this antisense oligo, even before we synthesize the molecule. So all these things can be done in silico and then can save a lot of time and money through artificial intelligence. As of now, in literature, there are at least 370 known diseases that can be treated this way because those are 370 diseases caused by errors in splicing and alternative splicing, even though we believe there are many more to be discovered. Some examples are cancers like myelodysplasia, AML, then genetic diseases like ALS, cystic fibrosis, MDF, DSMA, and those are just examples. So in the years we've been around, we're having a lot of traction. We are running 4 pilots with pharmaceutical companies right now. Well, actually 3 of them are ongoing, and 1 is already done. We have confidential agreements with 3 more partners. We can't really disclose who they are because of confidentiality issues, but you can look them up. They're somewhere in these logos. Yeah, you know, guess. But what I can say is we've talked to all these companies already, you know, and trying to make progress and collaborate with all of them. We have scientific partnerships with Memorial Sloan Kettering and Rockefeller to validate our predictions because everything we do is in silico and we need to make experimental validation. So that's what we do with these partners. And one of our strategic partners is public, so we can say H3, subsidiary from Eisai Pharmaceuticals from Japan. They're the first, our first partner, which is already public, so we can talk about it. So the way we work with the partners, with clients or partners, we prefer to call them partners because there is a growing interaction for both sides, we think, is to, first of all, establish confidentiality. Second, sit with the client or partner, understand what they exactly need, how can we help them, and then customize our product to fit exactly their needs. So this usually turns into a pilot that takes between 1 to 3 months. We charge between 50 to 175K for the pilot. So now we have a prototype which is customized. We scale it up, deploy, and then go to a licensing agreement which we're negotiating. We're looking to make 250, sorry for the messages in the middle, 250K for a 6-month license for about 1,000 input data sets. But the biggest challenge for us as a company is to promote our own drug discovery. We're internally looking for drug targets for ALS and immuno-oncology. And we look in the future, once we make interesting discoveries, to leverage our pharmaceutical partners to take these findings into the clinical trials faster, which we, of course, will validate with our academic partners that I mentioned before. This is to show that we have an amazing board. people in this, Adrian, I showed his photo, he was my boss. Michael Zhang, he also was my boss. He used to be here at Cold Spring Harbor, now he's at Dallas, and she won university in China. For example, Adrian and Tom Tuchel, they are experts in RNA. Adrian in splicing, Tom Tuchel, he's the inventor of the PAR clip technology to map the position of the RNA binding proteins on the RNA. And Mike Zhang and even Mike Schatz, three, probably you all know them, big people in genomics, and they're all helping us, and that's great. And just to close, make an announcement, we're closing a $1 million financing round right now. Dolby Ventures and Dynamic are already in the round. We're looking for more investors to close, hopefully next month. We are looking forward to do all these tasks, complete our commercial release of SpliceCore, scale the business, establish most of the relationship with pharma and so on. And also, most recently, we became a portfolio company from Breakout Labs. That's the Peter Thiel's Foundation for Life Science. That's separate from this round. So it's been a very exciting year for Envisagenics. We're hiring, we're growing, looking for bioinformaticians. So if you know any, let us know. And we'll be happy to take questions in the break. Maria and I are leaving today 2:30. We had to catch a flight, but we'll be happy to talk to anyone. Thank you.